Good day everyone, my name is Jeremy Hai and today I'll be presenting our work submitted to the ISMRM 2024 in Singapore. So the goal of this research is to investigate whether the nuclear Overhauser enhancement effect at negative 1.6 ppm can uncover new insights into ischemic injury. In an ischemic stroke, vascular occlusion occurs in which the blood flow to the brain is either blocked or severely restricted. This triggers a complex series of cellular events known as the ischemic cascade. When blood flow to the brain is disrupted, the affected brain cells are deprived of oxygen and glucose, leading to a rapid depletion of ATP since cells switch to anaerobic glycolysis, which is not as efficient as aerobic glycolysis. Due to lack of ATP, the sodium-potassium ionic pump will fail, leading to anoxic depolarization, which will then cause the rapid release of the neurotransmitter glutamate, which will stimulate the influx of calcium ions into the neurons. High intracellular calcium levels do two things: cause mitochondrial collapse and generation of free radicals. Free radicals will cause oxidative damage to the cellular structures, including proteins, lipids, and DNA, and a collapsing mitochondrion will release more free radicals and apoptotic factors. This vicious cycle continues till the cell membrane undergoes lipid peroxidation and the cell dies. Previous studies have linked NOE at negative 1.6 ppm to choline-containing phospholipids, and since the cell membrane is made of phospholipids, NOE imaging at negative 1.6 ppm can be a possible biomarker to identify the tissue that is affected by lipid peroxidation and at immediate risk of cell death. The potential of NOE imaging at negative 1.6 ppm for stroke diagnosis was investigated in an animal model, rat model. Diffusion-weighted imaging, perfusion-weighted imaging, CEST, and T1 data were acquired from 5 rats that underwent MRI at 9.4 Tesla after middle cerebral artery occlusion. The apparent diffusion coefficient, cerebral blood flow, apparent exchange-dependent relaxation AREX at 3.5 ppm, and NOE at negative 1.6 ppm were calculated. The binary masks of the segmented ADC, CBF AREX, and NOE areas were overlaid to produce the mismatch maps of the deficit areas. To quantify the APT signal at 3.5 ppm, these equations were used, where delta omega is the resonance frequency of the labeled proton, in this case 3.5 ppm, ZREF is the reference signal at negative 3.5 ppm, and T1 weighted is the water longitudinal relaxation time. Now, quantifying the NOE effect at negative 1.6 ppm presented a unique challenge due to its location around the shoulder of the Z spectrum and its relatively small magnitude compared to other effects. Direct observation of the Z spectrum often doesn't capture it accurately. To address this, a novel approach as shown in this equation was implemented. The main purpose of using 19 ppm over 13.5 ppm is to get a reference that is free from magnetization transfer or empty effects, NOE, and any other exchanges. The signal is too small, and we need a unitless reference so that we can quantify the small NOE effect better. The opposite NOE offset at 1.6 ppm was not used as reference, as it may have contaminations from nearby CEST effects, such as amine around 2 ppm. So for the results, the study found that the NOE signal was lower in the brain region affected by the stroke compared to the healthy contralateral region. This suggests that NOE imaging at negative 1.6 ppm might be sensitive to changes occurring within the ischemic tissue. Interestingly, unique patterns when comparing NOE with AREX were observed. While both signals showed decreased intensity in the stroke region, their spatial distribution differed, suggesting a unique sensitivity of NOE to changes in the ischemic tissue. This figure displays non-CEST MRI data from a representative animal. The apparent diffusion coefficient decreased and T1 hyperintense in the ischemic area, which is consistent with previous findings. As for the CEST data, a dip was observed around negative 1.6 ppm, and this dip suggests potential changes in the choline-containing phospholipids in the cell membrane, which could be associated with lipid peroxidation. The NOE deficit area was hyperintense and coincided well with the ADC deficit area. So as it can be seen here, the AREX deficit area and the NOE deficit area coincided well with the ADC deficit area. The NOE deficit area was equal to or larger than the ADC deficit area but smaller than the AREX. This is probably due to the sensitivity of each modality to different changes or stage of the ischemic cascade. The next figure combines data from multiple animals. Here we see maps for ADC, CBF, ARX, and NOE. The final image shows a mismatch map, where different colors represent the different imaging techniques and their respective deficit areas. The difference in size suggests that each imaging modality might be capturing information on a different stage in the ischemic cascade. So for DWI, it's representing the irreversibly damaged tissue. NOE represents the lipid peroxidation at risk tissue. APT, acidotic at risk tissue. And PWI shows the benign oligemia. These results demonstrate that NOE imaging could provide valuable insights and has potential to improve stroke diagnosis by offering a more comprehensive picture of the ischemic cascade. So in conclusion, using CEST coupled with DWI and PWI, the ischemic tissue may thus potentially be separated into 4 zones: the ischemic infarct, at-risk tissue undergoing lipid peroxidation, acidotic at-risk tissue, and the benign oligemia. This helps to better understand the pathophysiology post-stroke and improve ischemic tissue fate definition. And while this study provides valuable insights, further studies are still needed to elucidate the origin and observe changes in NOE at negative 1.6 ppm and to establish a reliable method for quantifying this small signal. Additionally, it's worth mentioning that the NOE effect at negative 1.6 ppm may not be visible with clinical scanners at 3 Tesla or below due to its small magnitude and proximity to the water frequency. However, advancements in ultra-high magnetic field clinical MRI, particularly at 7 Tesla, offer promising avenues for investigating the NOE effect at negative 1.6 ppm at the clinical level.